Hello.
Okay. So, hello and good evening to all the students. Today we are going to talk on the week 11 class on the course of experimental biochemistry, which is being taken by Professor Shagata Dasgupta and Professor Shomodaya of IIT Kharagpur. So today is the week 11 live class session. So like I always do, first I will be talking about the lecture in brief, and then we will be solving a few question and answers. Okay, so I am sharing my slides with you all. I guess my slides are visible to you all. Okay, so in today's class we would be mostly talking about protein ligand interactions. So in the previous classes we had already discussed about a lot about proteins, the purification of proteins. So now it's time where we need to do a few interesting functional assays related to proteins. One technique that we use, or one thing that we do with proteins, is to check its interaction with other proteins or macromolecules, which we also call as ligands. So these ligands can be another protein, or carbohydrates, or your even small molecules, even ions. Okay, so today's class will mostly revolve around the protein ligand interactions. So the binding of the ligands to a macromolecule. So the secret of life is the molecular recognition, that is the ability of one molecule to recognize another through the weak bonding interactions. This was said at the 25th anniversary of the Institute of Molecular Biology at University of Oregon by Linus Pauling. So binding is the first step that is very important for our biological response. Before macromolecules can perform a function, they normally just interact with a specific ligand. In some cases, like myoglobin, binding and subsequent release of the ligand might be the sole function of the macromolecule. To understand the binding, so to understand the binding, we have to consider the equilibria that's involved, how binding is affected by the ligand and macromolecule concentration, and how to experimentally analyze and interpret binding data and the binding curves. So reversible binding of ligands is very, very important, specificity of ligands and the binding sites. The ligand binding is often coupled to the conformational changes, sometimes quite dramatic, induced fit. In multi-subunit proteins, conformational changes on subunit can affect the others. That's via cooperativity. Now, interactions can be regulated. So, the interactions with the other molecules that can be reversible, can be transient. Process of chemical equilibrium, like you have A plus B reversible with AB. So molecule that binds to a protein is called a ligand, which can be a small molecule, which can be proteins, carbohydrates, lipids, fat structures, can be anything, but we call them as ligands, where it binds to the binding site of the protein. Ligands bind via some kinds of non-covalent forces that dictates the structure of proteins as well as function, and it allows the interactions to be transient in nature. Now, binding is has a cooperative description, that is considered a process in which the ligands binds reversibly to the site of the protein. The kinetics, sorry, the kinetics of such a process is described by via the association rate constant Ka, or the dissociation rate constant, that is the Kd. Okay, now after some time this process will reach a equilibrium, like for a chemical reaction or something, where the association and the dissociation rates are equal. You have Ka multiplied by the concentration of the protein and the ligand, which is equal to the Kd of the protein ligand. And the equilibrium composition is characterized by the equilibrium constant, where you have Ka, that's the chemical equilibrium constant, which is equal to the concentration of the protein ligand complex divided by the uh, concentration of the protein and the ligand individually, which is equal to your small Ka by Kd, that's the equilibrium composition. Now, when we consider binding in terms of the bound fraction, we have to analyze it. Now, in practice, what we do is we determine the fraction of the occupied binding sites by theta. So, theta is your total protein ligand concentration divided by the protein ligand complex concentration plus the protein concentration. That's the total number of the fractions of the occupied binding sites in the protein. Now, when we substitute this with, so we get back to the equation from where, sorry, so we get back to the equation from where we had started. That's K equals to PL by P into L. So you have, we substitute this PL with this term, and so theta can be simplified and written as the concentration of the ligand divided by the concentration of the ligand plus 1 by Ka. Now Ka is the association constant, but 1 by Ka is the dissociation constant. Okay, so dissociation constant, so we can simplify and write the fraction of the occupied binding sites in the protein is actually equal to the total concentration of the initial concentration of the ligand divided by the initial concentration of the ligand plus the dissociation constant value. If we know theta, the initial concentration of the ligand is previously known, we can calculate D. And this Kd value is very, very important because that determines the affinity, that will tell you about how strong your interaction is, or how well the protein binds to the ligand. So we can talk about Kd in the terms of a graphical analysis, where the fraction of the bound sites depends on the free ligand concentration and the Kd value. So experimentally, ligand concentration is known. Kd can be determined graphically or via least squares regression. Okay. Now this equation is very, very important, where you have Kd equals to, so it's 1 by Ka. So Kd equals to concentration of P into L divided by the complex, which is equal to your total protein concentration minus the bound, the complex concentration into the ligand concentration divided by P and L. So simplifying and plotting the graph, you get an inverse plot where you have plotted the complex concentration divided by the ligand concentration, and in the x-axis you have is the complex concentration. So PL max divided by Kd, so slope is minus 1 by Kd. From there you can calculate the dissociation constant. And if you want to know about the the maximum complex concentration, you can actually get it from the intercept of the x-axis. Now, uh, when we are talking of all of this binding, we have talked about the kinetics. Now the thermodynamic parameters have to be also understood. So we need to build the thermodynamic connections of the binding of a protein and a ligand. So the interaction strength can be expressed as association constant, which is molar inverse. Then you have dissociation constant, that is in molar, interaction free energy delta G. So all of these can be calculated. So delta G equals to delta H minus T delta S. That's the Gibbs free energy equation, from where you can get an idea about the interaction binding, or whether the reaction is feasible or not. And then delta, so K equals to PL divided by P into L. So the relationship is minus delta G equals to minus RTL in Ka, which is equals to RTL in Kd. So your delta G equals to RTL in Kd. So we need to note that lower the Kd value, higher is the affinity for the protein. For the ligand, higher the Kd value, like if you have in millimolar or maybe even more than that, then that is has the lowest affinity of the protein for that particular ligand. So strong binding is a Kd value which is less than 10 nanomolar, and a weaker binding is a Kd value which is greater than 10 micromolar. So if you see some protein dissociation constants like you have for avidin, biotin, or an insulin receptor versus an insulin, you can see where this is in femtomolar, you have this is around zero point 0.1 nanomolar, then another zero point 0.4 nanomolar, you have around say zero point 0.1 micromolar, okay, or 100 nanomolars, 3 micromolar, and 2 20 micromolar, something like that. So you can see the strongest binding is in the region of biotin avidin. Then in the mediocre ranges you have the antibody antigen, then the sequence specific protein DNA, okay, then typical receptor ligand interactions, and in the lower affinity you have the enzyme substrate. That is very very important, because you cannot have a very stronger affinity for an enzyme substrate, because the enzyme at the end of the day has to come off the substrate to give the product of your interest. So all of these affinities, the nature has designed these affinities in such a manner that we can utilize it in the different applications. Now the Gibbs free energy, that's the delta G, which is equals to delta H minus T delta S, and delta G equals to RTL in Kd. So the binding constant can be connected to the Gibbs free energy of the binding using the following equation. Now binding constant of Ki 10 to the power minus 9 corresponds to the Gibbs energy of around 53.4 kilojoules per mole at body temperature. Change in Ki of one order of magnitude means a change in the Gibbs free energy of 5.9 kilojoules per mole. So you see this is the binding equation which we mentioned, and this is the delta G. Now you can write this equation in many ways. So there are important types of protein ligand interactions is through your hydrogen bonds. That's typical charge assisted. Then you have the ionic interactions because of the effect of the pK values or the local pH of the binding environment. You have several hydrophobic interactions, cation pi interactions, okay, due to which the protein ligand interactions actually happen. So the hydrophobic interactions are mainly caused via the displacement, or more correctly put, the liberation of the water molecules from the lipophilic environment of the binding pocket. Right? Great. Now we need to find about a few techniques which we want to use. Okay, so there are a few techniques that needs to be used for understanding the protein ligand constant, the interaction. So the first one is the isothermal titration calorimetry. That's a direct method. It measures the heat change associated with complex formation at a constant temperature. So the complete thermodynamic profile of the molecular interaction can be understood in a single experiment. You have enthalpy, the entropy, the Gibbs free energy, the binding affinity, Kd values, the stoichiometry of binding. All of these details can be found out from one single instrument. So that's so what happens in an ITC. So you have a syringe over here, you have the adiabatic jacket that measures the change in the temperature. So you want to use it to so this um, so it keeps delta t equals to 0. So to maintain delta t equals to 0, you have the reference cell, the sample cell, and the constant power to the reference cell heater. So any change in the heat will be measured in the detector. So you see, you get graphs like this. With time you see graphs like this, and upon so endothermic heat event, so this where you have positive values. Sorry. So you see the graph over here. Yeah. So it's a curve like this, and on the other hand, a curve like this indicates an exothermic heat event. Now, so that's the same thing. The sensor, you have the lead screw. So here you are measuring the temperature. So you inject your sample over here, plunge it, and string the syringe. This is the outer shield, the inner shield, the reference cell, and the sample cell. So you can see over here. So this is the review of the free energies, enthalpies, and the entropies of binding. So R is given, T is given, and now we need to calculate a few things. So in the convenient rule of a thumb, a tenfold change corresponds to 1.4 kilocalories per mole. So how many kilocalories per mole change in free energy we need for a kd equals to 100-fold? So that's minus 2.8 kilocalories per mole, because it's log 2. So log 100, that's 2. So for tenfold change, you see 1.4 kilocalories per mole. Okay, so delta G must be large and negative, delta H for that. So how can an interacting be strong or the reaction to be feasible? So we have to have delta G must be large and negative, delta H must be large and negative, and delta S must be large and positive. Okay, so that is about the ITC experiment. Okay, so the next one is SPR. So that's surface plasmon resonance. So what is surface plasmon? So longitudinal charge density wave along the interface of two media, where one is the metal, other one is dielectric. So the principle is that SPR is excited by a depolarized total internally reflected light at a glass metal film interface. The surface plasmon enhancing the evanescent field amplitude, that is E. Now in biocore systems which use a sensor chip, this interface takes the form of an exchangeable gold coated glass slide. Its sphere is observed as a dip in the reflected light intensity at specific angle of reflection. So a linear relationship is found between the resonance energy and the mass concentration of biochemically relevant molecules. So the typical SPR signal is where you have the light source, the plane polarized light, and its reflected light. So you have the prism over here for the optical detection. Then you have the sensor chip over here. So you have the proteins bound to this sensor chip and you allow the ligands to flow over here. So first you have is the binding and then you wash it to allow the dissociation of it. So this is the binding curve, this is the dissociation curve. So from there you can actually analyze the data and get an idea. So the physical applications is that it measures the dielectric properties, adsorption processes, surface degradation or hydration of polymer films, the thin organic monolayers or bilayers, and the biological applications as biosensors for specific biological interactions including adsorption and desorption kinetics, antigen antibody binding, and epitope mapping for the determination of biomolecular structure and interactions of proteins, DNA, and viruses. Then you have the lipid bilayers, non-specific biomolecular interactions, biocompatibility, followed by tissue engineering. Then the few advantages of this surface plasmon resonance is that it has the ability to perform real-time measurement. You can calculate the binding as well as the dissociation in the real time, and it's insight to a dynamic nature of binding system and layer information. Use of selective slides to study the binding events and to eliminate the need of labeled reactants, and it's exceptionally sensitive with small quantities of purified reagents are required. However, there are a few disadvantages. That's the lack of sensitivity while monitoring low molecular weight adsorbents, the rate limiting factor of mass transport affecting the kinetic analysis, and so the methods that we can use to improve the sensitivity is coupling to an atomic force microscopy or mass spectrometry. The another very interesting technique is the electrophoretic mobility shift assay, also known as the gel shift assay or the gel mobility shift assay. There are lots of names have been given to this EMSA experiment. That's the electrophoretic mobility shift assay. It's a technique to detect whether the protein is attached with the DNA and an RNA or not. So as we know, there are many cellular processes where protein needs to interact with the DNA or RNA, like in transcription or translation. It's originally utilized broadly in the investigation of sequence-specific DNA binding proteins, such as the transcription factors. IMSA has been additionally developed to explore DNA protein interactions, RNA protein interactions, and all. And it's a very sensitive method using radioisotopes to label low nucleic acids and autoradiography. It is possible to use very low concentrations and small sample volumes. So the purified protein and the cell crude extract are incubated with an isotope labeled DNA or an RNA probe, and the complex and unbound probe are isolated on a denaturing propylene gel electrophoresis. That's a native page. So the mobility shift assay is the electrophoretic separation of protein DNA or protein RNA mixture on a polyacrylamide or agarose gel for a short period. The speed at which different molecules move through the gel is determined by their size, charge, and sometimes the shape. So how do we do the titration? So we have a certain concentration of DNA, we add it in all of that, followed by increasing or decreasing concentration of the proteins, and then you will see that there is a lane that is for the free DNA. So the lower is for the free DNA, the above lane is for the protein ligand or the protein DNA RNA complex. Okay, so with the increasing concentration of the protein, you will see that that your protein interacts more with the ligand upon increasing the concentration, and after a point of time it reaches the saturation. So you see over here the DNA bound to the two proteins, then DNA protein complex, and this is the free DNA. So this is the free DNA and this is the protein DNA or RNA complex. Now from here we have to calculate the dissociation constant. So what is the dissociation constant? Dissociation constant is the point where 50% of the DNA or the RNA, whatever you are using, is bound to your protein. So 50% when the ligand binds to the protein is where we need to get the is where we get the KD value. So the applications of EMSA is that we can study conformational changes in DNA, for example, an EMSA 2D variant with electron microscopy for characterization of the conformation, then analysis of cellular extracts for the presence of certain DNA binding proteins. Bound and free DNA are separated using EMSA. It's useful for stoichiometric analysis. Number of proteins that bind per DNA fragment can also be calculated. It is also used to study whether the transcription factor is attached to the DNA or not, if it's attached to a targeted or not in our functional assays or activities. Okay, so the other advantages of EMSA is that it can be used with a wide range of nucleic acids and different sizes and structures. It does not matter. So a wide range of proteins can be used with a wide range of DNA size of the DNA. As long as it's fluorescently labeled, it does not matter. And it's relatively a simple method to perform, used for qualitative analysis, useful for the detection of protein that binds to the DNA and cause difficulty in migration of fragment on the gel. It's a highly sensitive method and is usually of low cost, so no special equipment is needed, because all you need to do is run a native page and just watch it under some kinds of luminescence. And in this technique it's possible to use both crude extracts and the purified proteins. Now, a few drawbacks that EMSA has is that it does not provide a straightforward measure of the weights of the proteins, as mobility is influenced by several other methods. There can be multiple nucleic acids that can bind to a DNA. There can be multiple binding sites of the protein where it can bind. So there can be the stoichiometry might change, so the size cannot be said properly. It's not an appropriate method for the kinetic studies. And dissociation is also one of the drawbacks of EMSA that can happen with time during electrophoresis and can prevent the detection. And lastly, interacting biomolecules must have certain or different electrophoretic mobilities to identify them and isolate them. Otherwise, if they're moving in the same region, then it becomes quite tough. Okay. We have another experiment like the fluorescence anisotropy, which we will be discussing shortly. Okay, so the last experiment, that is fluorescence anisotropy. So before that, let's understand the polarization in a fluorescence experiment. So we have the excitation over along the x-axis and the detection along the y-axis. So y is the total intensity, IT equal to I of parallel plus 2I perpendicular. So you see along two axes it gets perpendicular, and along one axis where you have the excitation, that's parallel. So for the reason of symmetry, the component polarized along x direction and y direction have the same intensity, but along the given geometry it can detect only one polarized light. So that is why it's one intensity parallel plus two perpendicular. So far, so we have considered the frozen molecules. However, in reality, the molecules are mobile and their rotation changes the orientation of the dipole theta. So changes along theta are so far. So changes in theta reducing the anisotropy that's caused by photo selection. So this is the equation. So where rho is the rotational correlation time, that's d by rotational relaxation time, which is the time for the given orientation to rotate through an angle given by the r cos e to the power minus 1, 68.42 degrees. Now for a spherical molecule, rho equals to uh, viscosity into velocity divided by RT, which is 1 by 6 of D. That's the translational diffusion coefficient. For a globular protein, you have viscosity into the molecular mass divided into partial specific volume plus degree of hydration divided by RT. So for the measurement of fluorescence anisotropy, so the plane defined by the direction of excitation and detection is called horizontal. The measured intensities are identified by two indices describing the orientation of the excitation polarizer and the analyzer. So we have to account for the different sensitivities of detection of the individual polarizations. So R equals to intensity of VV minus GIHV divided by I of VV plus 2 GI of VH. So G equals to SV by SH. So that's for vertical versus divided by the horizontal. So that's the time resolved fluorescence anisotropy. We have the photon counts divided by the channel. So the scattered light can cause higher anisotropy because it's completely polarized. It's necessary to minimize the scattering for the accurate anisotropy measurements. Now, polarization methods are suited to study the aggregation state of proteins. So it's considered, for example, the case of a protein dimer monomer equilibrium. So following either intrinsic protein fluorescence or by labeling the protein with suitable probe, one would expect the polarization of the system to decrease upon the dissociation of the dimer into the monomers, since the smaller monomers will rotate more rapidly than the dimers. So lower P, and here you have the higher P. So hence for a given probe lifetime, the polarization of a monomer will be less than the dimer. Okay. Now let's get back to the practice problems. Okay, so let us discuss them one by one. So the first question, for a binding reaction, Protein plus ligand is PL. So the unit of K on and K off have the units of dash and dash. So K on is molar inverse, second inverse, and your K off is only second inverse. Yes, that is correct. The next one, which of the following is a technique for studying DNA protein interactions? Which setup does it require to perform the experiment? You have surface plasmon resonance, UV visible spectroscopy, EMSA native page, EMSA SDS page, DNA agarose gel electrophoresis, urea polyacrylamide gel. So for DNA protein interactions, so we can use SPR but not this. EMSA native page, we can do this actually. EMSA and SDS page won't work, and agarose gel electrophoresis won't work with the urea polyacrylamide gel. So here also B is the correct option. So the next one is from which of the following experiments can you determine the binding kinetics? Under favorable conditions. So is it EMSA, ITC, fluorescence anisotropy, or SPR? So 1, 2, 4 are correct, but 3 is incorrect. Okay, so let us see which of the following experiments can be used to determine the binding kinetics. So you want to check the binding kinetics. Okay. So from where can you check the binding kinetics? So EMSA you can actually do, you can check the protein DNA or the protein ligand binding over when run on a native page. ITC with the heat transfer also you can get the binding kinetics, that's the K on K off. Fluorescence anisotropy will also do when you excite it and you do that. And SPR is also fine, so you have the proteins bound to the gold sheet and then you pass the ligand. So that's a very real-time experiment. So all the options are correct. So option C. The next one. Enthalpy of a binding reaction can be determined from. So enthalpy of a binding reaction. So you need to find the enthalpy. Yes, so enthalpy can be found, or the thermodynamic parameters can be found very well from the ITC experiment. The dissociation rate constant K off for a protein DNA interaction is determined by EMSA. The intensity of the free ligand can be fit into which of the following equations? So that's a tricky question. So it's intensity which is equals to your initial intensity into 1 minus e to the power minus k of t. So kt, e to the power minus kt. So over here kt will be your k of, to obtain the k of equation from the IMSA experiment. So you see a curve over here. So the total heat can be expressed as, so this is all of this is Q1, Q2 and up to Q5. So what's the total heat? Is it a multiplication of all of that? Is it a summation or the average divided by the Q5, or is it of the only best 4 of it? Yeah, so the total heat is the addition of all of the heat generated. So it's Q1 plus Q2 plus Q3 plus Q4 and Q5. So that's the total heat over time. Now, which of the following statement about protein ligand binding is true? The Ka is independent of such conditions as salt concentration and pH. When all the binding sites are occupied, the Ka equals to the ligand concentration. If Ka is increased, then the binding will be tighter. The larger Ka means weaker affinity. So let's go one by one. The Ka is independent of conditions as salt and pH. That is not correct. When all the binding sites are occupied, the Ka equals to the ligand concentration. That is also not correct. If Ka is increased, then the binding will be tighter. This is the correct option. And larger Ka means weaker affinity. That is incorrect. Larger Kd means weaker affinity. The next one, a cytochrome P450 enzyme binds with a newly synthesized drug with a Ka of 10 to the power 7 molar inverse. What is the most likely measured Kd in the presence of a competing inhibitor? So ideally what is supposed to be the Kd, that is 1 by Ka, that is 10 to the power minus 7 molar. So that is supposed to be the answer, but in the presence of a competing inhibitor, what happens? The affinity decreases, and the affinity will decrease and it will become this one. So 10 to the power minus 6, because the other options, it does not become 10 to the power 6. Okay, it's a huge change. So it has to be 10 to the power minus 6 or 10 to the power minus 5. So 10 to the power minus 6 molar will be the Kd in the presence of a competing inhibitor. Okay. The next one, you have ITC, fluorescence anisotropy, SPR and the IMSA. So you have to match the following. So which one is correct? So let us see ITC. So let's start from the description. It's a resonant oscillation of the conduction of electrons at the interface between the negative and the positive permittivity material stimulated by incident light. Yes. So that one is fluorescence anisotropy. Thermodynamic parameters of interaction in solution. So that is so 4 is 3. Yes. So this one is SPR. So 1 goes with 3. So 3 is 2. And you have EMSA experiment, that is to determine the protein complexes with nucleic acid. So 4 is 3. So yes. So D is the correct option. Now if the free energy delta G for the binding interaction is known, then we can calculate. So if delta G is known, what you can calculate? So you can calculate the association constant, you can calculate the dissociation constant, the kinetic parameters, that's k on and k off, cannot be calculated. So C is the correct option. Now the k on and k off values for the ligands X and Y of the enzyme is given. So from this, what can you tell? X binds E more tightly, Y binds E more tightly. Xe complex is longer lived than Ye. Ye complex is longer lived than Xe. So what we know, so K on is 10 to the power 4. For Y it's 10 to the power 7. And for K off it is 10 to the power minus 3 and 10 to the power minus 1. So this value is more. So Y binds the enzyme more tightly. From K on we can tell that. And what we can tell that Xe complex is longer lived than Y complex, because the K off is more, or is very very less for the Xe complex. So 2 and 3 are the correct options. Number 12. In ITC experiment, for a titration experiment to determine the dissociation constant, it is necessary to vary the temperature. The ligand concentration is varied and protein concentration is kept constant. Protein concentration varied and ligand concentration kept constant. The concentration of free ligand, free protein and bound complex must be determined. So for a titration experiment, an ITC, it is necessary to vary the temperature. This is not correct. The ligand concentration is varied and the protein concentration can be kept constant. And also the protein concentration is varied and the ligand concentration can be kept constant. So these two are correct. So option B, that is 2 and 3 are the correct options. The next one. Our researcher designed a new drug and wanted to study the interaction of this drug molecule with the peptidase enzyme. If the drug molecule complex dissociates with an off rate of 149 second inverse, then what's the time constant for this reaction? So what's the time constant for the reaction? So how do you calculate it? So it's K of is equal to 1 by tau, which is or tau is equal to 1 by K of. So tau is equal to 1 divided by equal to this one. Okay, so the dissociation constant for a protein DNA interaction is determined by EMSA. The intensity of the free ligand at a corresponding time interval is given, so you have to determine the K of. So what you need to do for this is that you need to plot the intensity versus the time starting from 1 to 60 minutes, and from there you have to find out the point at which the DNA binds the time at the point where 50% of the DNA is bound to the protein, so that's your KD, and from KD you have to calculate the K off, and the equation will be 0.1 minute inverse. A very similar protein ligand titration experiment was repeated where the protein concentration of 10 micromolar is used, which is more than the expected KD. So the concentration of the ligand and the protein ligand complex for each titration point is given. So you have to determine the dissociation constant or KD. So similarly, you have to plot PL by L versus. So from here you have to calculate KD. So let's get back to the where we had talked about KD, the equation and all. Yes. So you see over here PL by L versus PL, so you will get a negative curve, and from there you can minus 1 by KD, so the slope, so it's a negative slope, so from there you can get 1 by KD, and from there you can calculate the KD. So all of this you have to ideally plot over there. You need to plot the curve, you can do it in an Excel file, and from there you can get the KD. So the slope is basically 1, so it will be 1 micromolar. Okay, yeah, so that's all from my part. Okay, so thank you all. We will meet again in the next week. Okay, and I will be sharing the slides, and the video is being recorded, so if you have any problems you can ask me in the next class or can write back to me. I will be very happy to answer them. Thank you.